GPCR structure and pharmacology. She did a PhD in medicinal chemistry in Moscow State University. Then she did a postdoc in Toulouse at the Institute of Molecular Medicine with Daniel Fourmi and Bernard Maigret. From there, she went on to do another postdoc at the NIH in Bethesda in the US, and she worked with people like Stefano Costanzi and Ken Jacobson. So, continuing to build a very strong experience with GPCR. And she's now a reader in molecular modeling at the School of Pharmacy at Queen's University in Belfast. And her focus remains on GPCR structure, dynamics, and pharmacology from a computational point of view. And she will tell us more about that. So her title is on her slides here. So I think the floor is yours, Irina. Thanks again for doing that.
So thank you, Nicholas. So welcome, everyone. And so thanks so much, Nicholas, Sarah, for invitation. I'm really delighted to give a talk and present some of our recent research. And yes, you see the title of my talk. So I'm going to talk about dynamic structural basis for allosteric modulation and signaling bias in G protein coupled receptors, GPCRs. And my focus mainly will be on free fatty acid receptor 2. But before going to this talk, I would like just a few slides to show you to introduce what we do at School of Pharmacy at Queen's University Belfast in my group. So we are a computational group and we use molecular modeling to elucidate a novel pharmacology of drug targets. And as Nico said, our main focus on G protein coupled receptors. And we are currently focusing on the search of allosteric binding pockets and understanding bias signaling, target polymorphism. And so we'll leverage these molecular insights then to guide compound design and virtual screening. And together with different experimental groups, we tackle these questions on number of G protein coupled receptors as a main our focus. And also we have something research on enzymes and ion channels. And alongside of using available molecular modeling techniques and methods, we also try to develop molecular modeling protocols tailored to address these questions, as you see in our pharmacological questions. And in our research, we use computational techniques coming from different computational fields: biophysics, chemoinformatics, bioinformatics, and data analytics. And today, my focus will be mainly on the application of molecular dynamic simulations in the search of allosteric modulators, allosteric binding sites, and understanding GPCR activation and bias signaling. And so now I would like just to give you a few highlights and introduce you indeed G protein coupled receptors. And these are the largest membrane protein family and probably doesn't need much of introduction. So we have over 800 members, GPCR members, and here you can see phylogenetic tree. And in the past 40 years, GPCR were proved to be druggable. And as you can see here, we have over 500 drugs approved by FDA that target currently around 120 GPCRs and more than 300 in clinical trials. So indeed, a lot of success. And structurally, GPCRs, as you can see here, the cartoon, so it represents itself a helical bundle. So it has 7 transmembrane helices that connected via extracellular and intracellular loops. And the endogenous molecules bind to the central cavity, and the highlights of this group of membrane proteins that it can actually transmit the signal from the extracellular side to intracellular side from a variety of endogenous molecules and really broad chemical space. So from little ions, as you can see, to large proteins. And despite its success, and indeed, I guess, the golden ages of GPCR research and drug discovery now we have, and despite of the success, we have a number of challenges. And one of the challenges, indeed, to design a molecule that binds to the central cavity for many GPCRs. And one of the challenges that obviously this central cavity is highly conserved. And if you have a number of GPCR subtypes, so it's hard to get a very selective molecule. So, and therefore, we actually don't focus here on the central cavity, but we focus on alternative cavities. And so these are allosteric sites that can locate in different parts of GPCR. And so now we know allosteric modulators for a number of GPCRs, and they can obviously enhance activity of orthosteric endogenous ligands, or indeed they can activate receptor on their own. So that will be positive allosteric modulators or agopams if they can also be, activators on their own. And then, of course, there will be an opposite spectrum molecules that would inhibit GPCR activation. So NAMs, negative allosteric modulators. And attractive feature of allosteric modulators, of course, it will be potential for receptor subtype selectivity, given those sites are not so conserved. Yeah, another point here is that they also, obviously, allosteric modulators depend on the presence of agonists and can selectively activate certain signaling pathways. And here, just to remind you, so you can see that we have 4 major groups of G proteins: GI, GO, GQ, GS. And here you can see the cryo-EM structures and the GPCR with G protein. And also we can have a signal via recruitment of beta-arrestin. And indeed, of course, there are ligands that can activate all of them, and there are ligands that can obviously selectively modulate specific pathway. And this could be quite useful if we could actually have allosteric modulator that would selectively activate specific pathway. We could reduce side effects, and indeed, we will have a better tolerance in chronic diseases. Also, another point that allosteric modulators can modulate duration and intensity of a receptor response, which may extend the therapeutic effect as well. And despite these positive features, we have only 3 drugs approved by FDA, and you can see here. And most of these drugs, so far allosteric modulators, until recently, have been discovered by random screening, high throughput screening. So, and our mission, of our research, and indeed actually to think about whether we could come up with some, of course, rational approaches and knowledge-based identification of those allosteric sites and finding allosteric modulators. And now, actually, if we go back to 10 years ago, and as a computational chemist working on GPCRs, so, those times, 10 years ago, when basically we just run the molecular dynamics simulations of GPCR and whether it will be X-ray structure or homology model, and when we analyzed the trajectories of those simulations using a simple geometry-based cavity detection algorithm as implemented, say, in MD pocket, we often saw a number of putative binding pockets, and here you can see in this surface representation in different colors on the GPCR surface, and so very interesting pockets that at the receptor lipid interface. But those times, it was actually difficult to imagine whether a small molecule drug can bind to those pockets, or it's just an artifact of, of simulations of algorithm in detection of those cavities at receptor edge. So, and now flash forward to today, of course, with all these achievements in structural biology, so X-ray crystallography, electron microscopy actually allow us to validate those pockets. And indeed, we have a number of cryo-EM structures with allosteric modulators. And here you see an image overlay of all those available structures of GPCR with allosteric modulators that I overlay to you. And you can see now just in the video that indeed allosteric modulators can bind GPCR in different locations at the extracell site, intracell site, and indeed in different parts of receptor lipid interface. So, and this is a really fantastic time, a wonderful time to actually start to use this data set, and to use computational algorithms to train based on this data set and come up with a protocol that would allow you computationally predict allosteric sites and indeed basically extend this set to all 800 GPCRs. And indeed beyond, so a GPCR could perfectly serve as a role model for other membrane proteins. And indeed, a lot of, actually not a lot, but a number of academic groups come up with different strategies how you can do it. And we also have our protocol, which I would like to introduce to you, which we actually use quite often in our GPCR research. So we recently published this protocol. It's called prop-confined dynamic mapping. And in this protocol, we use very old computational methods, co-solvent MD simulations. And what it means that we use organic solvents that obviously and simulate in the presence of organic solvents, and those small molecules can quickly diffuse into the protein cavity, identifying this cavity as accessible, capable to form interactions. So this approach was quite successful for mapping allosteric sites in soluble proteins, but you especially if you're a computational chemist, you could imagine it's maybe not so easy straight away to apply for membrane proteins on the simple reason that all those organic solvents can quickly diffuse to the protein, to the membrane and redistribute it, so we lose the sampling in our simulations. So what we propose the protocol that would allow you to use this method in the world of membrane proteins. So, and our protocol has two strategies. The first is that we introduce during simulations this cylinder wall potential. So here you can see our system setup. We introduce the box, and the box can be placed in different locations of the extracell site, as you can see here, or intracell site. So we prepare the system. During equilibration, we confine this props just in the water. We don't allow to interact with the protein. And during production stage, we extend the cylinder and we confine the movement of the probe within a specific area of the system. And here you can see this extracell site. So eventually you have a simulation that looks like this, as you can see here. So the probes are confined and they move and sample the area of the interest. And so there's more chance to see the events, and indeed it's you enhance your sampling. And another strategy over here, in addition to organic solvents, so we use fragments derived from the allosteric modulators to actually sample the area of interest. So this protocol has a number of advantages. So enhanced pocket sampling, as I already said, reduced sampling noise as well, and low computational cost. And so this protocol is available on GitHub, and everyone can use it and adapt to, I guess, different GPCRs, to different locations, and indeed different membrane proteins as well. So we validated this protocol in different settings, retrospective settings and prospective settings. And we looked at the cryo-EM structures, X-ray structures, homology models, and AlphaFold models as well. And we predicted allosteric sites for dopamine 2 receptor, CCR4 chemokinin 4 receptor, neurotensin 1 receptor, and the focus will be here on free fatty acid receptor 2. Okay. So you also see the pipeline of different computational programs that we use to actually, to set up and run these simulations in this slide. Now I'm moving to free fatty acid receptor 2. So this is, we have been fascinated by this receptor for a very long time and working closely with Professor Graham Milligan from University of Glasgow. renowned pharmacologist, and later on we continue working on this with Chen Zhang from University of Pittsburgh. So free fatty acid receptor 2, it's a group A GPCRs, and it's a promising target for metabolic disorders, obesity, diabetes, and inflammatory diseases, ulcerative colitis as example. And this receptor is activated by very small endogenous molecule like short chain fatty acids. So they're produced by gut microbiota. Yeah, and so we don't have a lot of ligands here as orthosteric ligands, only one agonist, TAC1375, and two antagonists. But what we have here, we have a number of allosteric modulators, positive allosteric modulators, so 3 distinct classes identified by random screening coming from 3 different companies, AstraZeneca compounds, Takeda compounds, and 4-CMTB from Amgen. Yeah, and what you also can see is that chemically they are different, and also you see that one of them, AstraZeneca, has a bias, so it only activates GI, but the other 2 activate GI and GQ, so they are unbiased. And so given that they were identified via random screening, so we don't know where they bind, and we have been working on this for quite a long time, back in 2011. So we tried to identify binding pocket for 4-CMDB, this Amgen compound, using homology models, and there was a lot of mutation made, but we didn't find the pockets. So yeah, and only in 2024, there was a, you actually, 2 groups, and including our group together with Chen Zhang, published a cranium structure of FFA2 bound to orthosteric ligands. And you can see fatty acid or indeed this synthetic molecule. So now this is a very good starting point to use this cranium structure to actually map the binding site for these molecules. So one point I want to make here is that from pharmacological studies and comparativity studies, what we know is that AstraZeneca and Takeda compounds, they don't cooperate. So it means they likely bind to the overlapping site. Yeah, and 4-CMTB cooperates, it can enhance efficacy or potency of those 2 compounds, AstraZeneca and Takeda. So it binds definitely to the different site. So, and now we focus on the first 2 molecules, given that, suggestion is they should bind to similar site. So we looked at these 2 molecules, and so we ran molecular dynamic simulations of this cranium structure that we have now. And with MD pocket, that I already mentioned, we identified several druggable pockets, as you can see here, shown with the surface, yeah. And then indeed we used our probe confined dynamic mapping to actually look at each of these pockets. And now I'll show you the fragments. Yeah, so the fragments that we know from structure activity relationship studies, that these are really a key fragments that we need to have to actually have activation of FFA2. So we got those fragments and we set up the simulations. So these are all atom simulations, but I show a very simple, obviously, video over here. And what was interesting to see is that these probes, yeah, so the probes form persistent interaction with this glutamate 106 at the receptor lipid interface on the intracellular side. And it's very interesting, especially for this probe, which is positively charged. It finds at home at the intracellular side at the receptor lipid interface. So that was very interesting, and this interaction is visible only in the MD simulations, and the simple docking to the static structure, cryo-EM, doesn't allow you to see this interaction because this glutamate, the side chain, pointed inside of the protein. It's not exposed to receptor lipid interface. So it again shows the importance of MD simulations in actually, sampling and finding those putative binding cavities. So based on these prop dynamic simulations, we suggested that indeed that glutamate 106 are important. And obviously the binding site might be in these locations, So then we turn to our collaborators, to experimental collaborators, to validate this. Yeah, and just last year, so Chen Zhang got the aquarium structures of these two molecules. And it's just like we predicted, the molecules interact with this glutamate 106. Yeah, so this glutamate, as you can see. So these are aquarium structures in the complex with GI, with a G protein, and with a GQ as well. So it's a definitely fantastic opportunity for us as a computational chemist to work with such a group with cryo experts, and indeed to validate our approach just in the best way, just not doing just a mutation, but indeed have opportunity to get cryo structures. So we then, of course, focused on the cryo structures to understand the, I guess, the binding interactions, and also understand the activation of these molecules and indeed biased signaling. So this slide shows you the snapshot coming from molecular dynamics simulations. And so within our simulations, that we see a stable and persistent strong interactions with this glutamate. Yeah, and also you can see I visualize amino acid residues around the ligand. Yeah, and so here you see that the color of the residue shows you the electrostatic energy. Yeah. So, and the thickness of the residue shows you the van der Waals interactions, as you can see. And this is a wonderful image to appreciate, what actually, who is the kind of important partner in holding these ligands that forms the stronger interactions, to see this hotspots. So, and indeed, glutamate is very important. And then you can see that there will be other interactions with serine 120 as example. And you can see also key interactions coming from hydrophobic aromatic residues. So, and if we just go back to this glutamate, so obviously we see the strong interactions, and we wanted to now understand so what's happening with this structure if you take out the ligands, yeah, and simulate just so to say empty form or without allosteric modulator. So, and what we've seen that in the simulations, so this glutamate moves again side of the protein and forms the sole bridge with arginine within the same helix. And this is all part of the famous E-dry motif. So this is conserved motif in the group A GPCRs, so actually responsible for activation. And what happens, this ligand does is just basically binds to this glutamate and kind of breaks this sole bridge. And we obviously simulated and looked at the dynamics of these interactions in different systems. And I just show a few of the systems for clarity of the slide. So whenever you take out the ligands, there's this beautiful violin plot. So you see that without ligands, even in the presence of agonists, you see the formation of the sole bridge, and the presence of the ligands, you see that indeed we have a high distance, so we have a breakage of these interactions. So again, shows the importance of this glutamate in holding the ligand, and indeed actually the suggestion that it's important also for breaking this sole bridge, which exists in the inactive state of a GPCRs. This is a conservative, as I said. So, and kind of explaining the activation part of this ligand. And so also we obviously wanted to validate these glutamates by just creating a mutants and mutation of this glutamate to glycine, abolish the activity of AstraZeneca compounds, Takeda 187, but still we have a binding of synthetic molecule activation still, and also for CMTB, which, as I mentioned, it's meant to bind to the different site. So from all this, what we could say that indeed E-dry motif can be targeted directly by small molecule ligand to activate GPCR. And in order to better understand this importance, and this is kind of novel aspect of this work, I'll show you next slide where you could see the common activation pathway of class A GPCRs. And we know that the activation of agonist, yeah, so on the central cavity, as I mentioned, so triggers conformational changes in the GPCR. And here on the left side, you can see the main kind of global change is outward movement of transmembrane helix 6 away. So you have this open state. But before actually this large movement, we have a number of micro switchers that basically, involved in these large global movements. And this will be conformational changes or rearrangement of contacts in the conserved amino motifs. Yeah, conserved motifs. So CWXP motif as a toggle switch, then PIF motif, hydrophobic motif. We are losing sodium ion in the sodium pocket during activation. Then there is NPXXY motif and E-dry motif just right on the intracellular side, close to the G protein, that actually the last stage of the activation. And what we see now with these two ligands, that it's not necessary, obviously, you can see now there's just outward movement in the pink, yeah, is like more in a structural way rather than the scheme that you see here on the left. And what we now learn from this multidisciplinary research, that you can actually target directly this motif on the intracellular side in a way without needing to interact with other parts of the receptor. So this is a really interesting discovery, and indeed shows you again that there are lots of druggable areas within the GPCR, and actually you could immediately target the key motif that basically regulates interaction with a G protein. Yeah. So, and now I think from that, I would like to move to this bias signaling, where it's really getting interesting. So these two ligands, as I mentioned, so one of them, so GI biased, so it only activates GI, but together molecule activate GI and GQ. So, and so we just then, collected all those cranium structures as a starting point. And what we have seen, and then we run the molecular dynamic simulations, we evaluated the angle of this intracellular helix 2. So those ligands, so apart from they interact with glutamates, they also have interaction with this intracellular helix 2. So yeah, with a loop. And in the case of AstraZeneca compound, so we see that the angle is a little bit different. So there is this upward shift of ICL2 and slight rotation. Yeah, and then of course there will be the question whether it's just because of the ligand or because actually we have a G protein, and we then run different simulations in different setup. And here you can see again another beautiful violin plots, and you can see different settings, obviously with orthosteric ligands, with allosteric, with GI receptors, and without GI, just only with allosteric. And then also you see complexes that come from different cranium structures of FFA2. Then we took this one of the structure that obviously doesn't have AstraZeneca compound, and we darken, we simulated. So we wanted to see the conformation of this ICL2, and we measure here this is simple measurements, root mean square deviation of ICL2 C alpha coordinates in relation to this cranium structure where we have AstraZeneca and GI. And what we see is that whenever we have AstraZeneca compound whether it's with G protein or just AstraZeneca compound, we see the deviation is very small. So it maintains this conformation of the helix. So G protein is not important. It's really more driven from AstraZeneca, from the ligands. And then if you actually dock this ligand to another cryo-EM structure, so you could see that the AstraZeneca holds and basically conformation similar to what we see in the cryo-EM structure with this AstraZeneca compound. So again, it just tells you that indeed, so the AstraZeneca stabilized this unique conformation of ICL2, and likely that this could be actually linked to this GI biased. So we also then wanted to characterize the conformation of this loop, ICL2 loop, and here you could see basically overlay where you see cartoon representation that will be cryo-EM structure, and this surface representation that's C alpha density of the carbon atoms, carbon atom density of C alpha atoms. So in the simulations, in the empty receptor, yeah, so you see basically, very dynamic loop, and so in the empty receptor, in the presence of orthosteric ligands also, we don't see this density. When we have a GI, so we already have stabilization, is quite rigid. In the presence of GI. And then also we looked at so this is ICL2 loop, yeah, and we looked also, I guess, with different ligands, like for CMTB, we looked at the compound 187 and 1729, and these are simulations without G protein, just in the presence of the ligands. And what we see is that AstraZeneca compound really kind of fixed the ICL2 in a specific conformation, while in the case of the Takeda molecule, so it also holds this alpha helix. It's very stable, but it kind of gives you a shift, as you can see, yeah. So, but both these two ligands, they stabilize, they regulate the conformation of ICL2, as you could see with all these different setup of simulations that we have made. And now if, highlights of ICL2. And now also interesting comparison, the same, so density coming of C alpha atoms coming from simulations and the cartoon representation cryo-EM. So if you take all these GQ complexes of FFA2 with different ligands, C4s, or the endogenous molecule, butyrate acid, synthetic molecule with different allosteric modulators. So we see that in different complexes bound to GQ, there is a very clear conformation, very similar conformation of ICL2. While if you simulate different complexes bound to GI, so we see variation of this conformation of the ICL2. So, and here you can see in the case of 187, so you see that, whether it's just a complex with GI, or indeed we also can dock to cryo-EM that didn't have this molecule, and we see that in both cases we have a conformation very similar, but again it's shifted. So without G protein, 187 stabilize a little bit different conformation, but it's still a rigid, quite rigid, I guess, position, not like in the empty state. So, and also, I guess another way to show this 3D representation, you just could basically go down and show in principal component in 2D representation. And here you can see 187. So we have cryo-EM with GQ, with GI, and without. And so in the case of the GQ, so in the pink, you see that we have a very distinct position. So for GQ, while obviously GI can form very different conformations and overlays also with I guess, simulations without GI. So from that, we could see that GI coupling allows for greater ICL2 flexibility compared to the GQ coupling. And so AstraZeneca stabilizes ICL2 conformation that favors productive coupling with GI over GQ. Okay, now just in a moment will be even more information. So this is a very neat, to my opinion, comparison. So simulating different scenarios, starting from evaluable cryo-EM structures, using cryo-EM structures of just FOFA2 and docking the ligands and seeing, the idea was again to confirm the contribution of the ligands to the stabilization of ICL2 and again show the importance of specific conformation of ICL2 in modulating specific G protein. So, and now from that, we started to look how ICL2 conformation interacts with the G protein. And the main contacts, direct contacts, is with the G alpha 5 helix. And so this is GI helix, you can see here. And then also at the bottom, when we have 187, also with a GQ. So ICL2 forms direct contact with alpha-2 GI helix or GQ. And so the main difference between GI and GQ is that we have contact with cysteine in GI and the tyrosine in GQ. So different non-conserved amino acid residues, yeah. And we then basically run different complexes, and here you can see all different complexes we run. We also run mutations, and we measured the distance between tyrosine of ICL2, yeah, just with some reference points, the closest contact, and then distance of basically from this residue. And so what we see is that, so the way how AstraZeneca stabilize basically the interaction with GAI. So it favors interaction with cysteine rather than bulk its tyrosine. And indeed, we could see like in the complexes where we don't have AstraZeneca, so the distance bigger compared to the complexes where we have AstraZeneca compounds. Yeah, so the distance is shorter. So basically, the way how AstraZeneca stabilize this conformation is basically, it just changes changes the behavior, the interaction with these non-conserved residues, and just basically gives you a steric hindrance with tyrosine. So, and indeed, if we would mutate this tyrosine in GQ to the cysteine that we have in GI, so we see that we can actually come back to coupling, and we can recover coupling of GQ, yeah, of GQ by AstraZeneca compound. So ICL2 conformations can regulate different G protein subtype selectivity via steric selection. So from that, what we can conclude. So it's a really a new way to look at this. It's a new mechanism that obviously here works to explain bias. And of course, one of the steps here is actually now to use these different conformations and think about if we can then design rationally a molecule that would bind to the specific G protein, GI versus GQ. And from pharmacology standpoint and physiology, here we actually need a molecule that activate GQ but not GI. So the other way around the bias. Yeah, so we have a AstraZeneca for GI, but we actually need GQ, and this is something that we want to do, and hopefully we will have some funding and we'll continue this work. And now I move to another molecule that I mentioned for CMTB. Yeah, as I said, that we tried to map the binding site of this molecule since 2011. We're not successful. So those times nobody even thought about that the molecules can bind somewhere else. So the idea was, okay, short chain fatty acids binds to the extracellular site, but there is extra kind of room, and maybe 4-CMTB can bind next to it. So, and then we mutated a lot of stuff around and residues around, and they didn't change the activity, so it still can activate receptor. So, and then only, I guess, at the end of last year, and we are in collaboration with Chen Zhang from Pittsburgh. So he got the cryo-EM structure of a 4-CMTB molecule, and it was fascinating to appreciate that this molecule binds at the receptor lipid interface in the middle of membrane. And here you can see there is only one polar contact with asparagine 230. Yeah, it's a very unexpected location, really superficial location near TM6 of FFA2 and single hydrogen bond. So we then wanted, of course, as a computational people, take this cryo-EM structure, try to learn more from that and explain the activation mechanism. And this is what we've done. So, so here you can see cryo-EM structure, and now you can see molecular dynamic simulations. And in the molecular dynamic simulations, we see a very interesting event. So now you will see it. So there is a flip and formation of the 2 hydrogen bonds. Yeah, and so actually the interactions will involve 2 hydrogen bonds. Yeah, 2 hydrogen bonds. So heterocycle is also engaged, and we have interaction with NH. And there is a non-classical, so to say, hydrogen bond between very polar chiral hydrogen and carbonyl group of this amide. Yeah, so this is a non-classical hydrogen bond. So that's very interesting. And of course, then the next question, can we validate this, binding pulse that already comes from MD simulations? And indeed, this is what we try to do. So our MD simulations, to conclude the slides, have provided a more detailed understanding of this binding pulse. And now let's look at the validation. The first thing that we wanted to validate, explain, is preference of S isomer, but not R isomer. And you can see the activities that are derived from original paper coming from Amgen. And so definitely a very big difference in activity. And what we actually see is that, of course, we don't have this non-classical hydrogen bond, which again, in this hydrophobic environment in the middle of membranes, it will be a quite strong hydrogen bond. And also, of course, there is a steric effect, this isopropyl group just basically hitting this helix 6 and the residue, this is isoleucine, very close. Yeah, and then also there is a shift with this phenyl group. So with this kind of network of interaction and the absence of non-classical hydrogen bonds and poor fit could actually explain preference of a specific isomer. So another thing, of course, we dived into structure activity relationship of 4-CMTB molecule. And so what this is the work coming again from our study in 2011. So what we see here is that if you have this hydrogen over here, an H, yeah, and you actually replace bimethyl, yeah, so you completely abolish activity, so it's inactive, yeah. And if you then modify the heterocycle, yeah, so a slightly different heterocycle, but then the nitrogen, this is Z nitrogen, if you put carbon, yeah, so you also completely get molecule inactive. So what it tells you from SAR, that these 2 nitrogens, I guess nitrogen and then hydrogen, are important, and they're involved in the hydrogen bond. So this is again the binding pulse that we see in MD simulations and we don't see in the cryo-EM. So indeed, again, shows the importance of MD simulations, that we definitely need to use it and validate all these static structures coming from experimentalists. And final validation from side of pharmacology, where we mutated asparagine 230 to aspartate, yeah, which actually can form 1 hydrogen bond. But what we see, we have completely loss of activity for this molecule. So it tells you that again we need to have 2 hydrogen bonds, yeah. So and it will not work. So all this supports the binding paths proposed by MD simulations. So, and this is indeed exciting news for us, for computational people, and building definitely confidence in what we are doing. Okay, so next thing was, of course, to understand how this molecule can activate for CMTB the receptor. And what we've done, so we've simply took out this molecule and we simulated the system without this molecule. And so what was interesting to see, this asparagine moves back to the protein, to this helical cavity, and starts to interact with the set of residues, as you can see here, and some of them coming from those conserved motifs, yeah, conserved motifs. And we run a lot of replicates, and we measure all those distances, and one of the distance of this asparagine 230 with aspartate over here, yeah, and there's different scenarios, as you can see, and this beautiful Weiling plots that basically show you that in the presence of 4-CMTB, we don't have this hydrogen bonds, but in the absence, we have this hydrogen bonds. So then the same way, if we look at this asparagine and then asparagine 265, the other one coming from this NPXY motif, again, in the presence for CMTB, we don't have these interactions, but when we remove, we have appearance of this hydrogen bonds. And so finally, we looked at the cysteine, and also we see that in the absence of 4-CMTB, so we see these interactions, and in the presence, we lose these interactions. So that's a very clear card demonstration of basically the importance of this asparagine 230. And then basically by looking at that and also exploring further, so we then looked at the hydrogen bonds between TM6 and TM7 cytosolic part. So, and you see this is, you can see here nomenclature in the GPCR world, which maybe I probably should needed to explain before. In GPCR world, so we have a nomenclature, as you can see here, and this is basically 7, as you can see, 7.49. So 7 stands for the helix, 7, and 49, it's basically the label of the residue coming from the most conserved residue, which is labeled as a 50. So it's basically next to a 50, so before actually 50, 49. So, and if you have this label, and you can easily navigate across all GPCRs. So it's just a unified nomenclature. It's quite easy labeling actually of GPCR residues. And when you look at the, again, hydrogen bonds between TM6, TM7, and we see that in the presence of 4-CMTB, we are losing those contacts. So, and also, for example, you can see there was a situation where obviously we run cryo-EM structures, then we docked 4-CMTB to another cryo-EM, we see the same. And also when we mutate asparagine 230, yeah, to alanine, just computationally. So, and then what we see that we are losing all these contacts. Yeah, and when we mutate to 30 to aspartate, we see the contact, because obviously aspartate can play the role in form of those interactions. So, and this kind of in this looking at all those simulations in the presence, in the absence, we propose this non-canonical activation mechanism that 4-CMTB binding disrupts hydrogen bond network and allows this TM6, TM7 split. And this TM6, as you see, and I show in the diagram, this large outward global movements outward from the helical bundle. And 4-CMTB partially can stabilize this active conformation even without G protein. Yeah, so it's just a very strong contact, and obviously the moving asparagine away from those interacting partners and activating receptors just allows to move helix 6 easily away. And in the context of activation pathway, again, I put this back slide. And as I mentioned, we have all those conservatives, microswitches that lead to the global movement. And now we are talking about binding sites that in the middle of membrane and a little bit indirectly involves those conserved motifs. So at the big kind of more close to the extracellular side and intracellular side. And basically, this is where we are targeting with a positive allosteric modulator the area, and we are getting activation. Yeah, so that's also a quite interesting observation, and indeed kind of non-canonical, unusual, where you could actually interrogate this area in the middle, and you don't need probably the whole pipeline. So, which is again needs to be validated, and we are planning to do it. Okay, so to summarize this story, as first that we have this probe-confined dynamic mapping protocol that allow you to map GPCR allosteric sites. And second thing, of course, that hopefully with this story, you could see how important molecular dynamic simulations in looking at the structural data, and indeed a more detailed picture of ligand binding. We can clarify a lot of aspects. And this is great news for computational people. And indeed, we propose two receptor activation mechanisms. You know, when you can directly target those buried kind of conserved motifs, E-dry on the intracellular side, or indeed in the middle of membrane by small molecule ligand, by a drug, and activate the receptor. So this is a really great. And great because in a way, it's quite often it was easy actually to discover inhibitor, right? So which means like negative allosteric modulator, NAM, right? So here we actually, by basically finding those pockets and looking at those interactions, targeting directly important motifs, we could actually activate the receptor. So it's a really interesting observation and quite novel. So we basically compiled this long story and got just paper accepted in Nature, which is just really great for us. And indeed, ICL2 loop, which obviously has different conformations and depends what kind of conformation we could stabilize by a drug. We could potentially design a molecule that will selectively signal via specific G protein. So this is also quite a novel concept, and we're going to use all this. Again, we continue working on this, and we try eventually our perspective, our future thing, whether we can use all this information coming from molecular dynamic simulations and indeed cryo-EM structures for designing rationally knowledge-based biased allosteric modulators. And as I mentioned, that what we want here in this receptor is to get GQ activators. So, and obviously collaborations and findings. So I'm definitely grateful to my experimental collaborators. So definitely allow us to bring computation in such a high level. Yeah, so we could obviously see how it all works and validate our hypothesis. So Professor Graham Milligan, pharmacology, and Professor Chen Zhen, cryo-EM structures, and my wonderful PhD student who ran all these simulations, Akim, and he's going to have a defense, Viver, just at the beginning of June. So, and hopefully it will go well. So PhD and a Nature paper. So it's definitely, hopefully, it's all sealed for him. And finally, I would like to say, so it's an industry workshop, and indeed, industry talks. So we definitely have industry partnership and collaborations, and we provide just GPCR drug discovery services, and either it will be just multi-year partnership or indeed flexible engagements, models like with the 3 months projects, or like ongoing support as well, and the core expertise. So obviously I mentioned already different techniques, and just here show that's something that we just really run on a regular basis and know, so to say, by heart. MD simulations, docking, virtual screening. We're introducing machine learning. So of course, supporting MedChem and looking at the QSAR, building QSAR models, pharmacophore design, understanding species differences, and indeed our specialized expertise and our sense of biosignaling and analysis around this, understanding agonist versus antagonist, and looking at the allosteric sites. So you can see the deliverables that we usually propose to industries, and indeed this is all hopefully quite successful, and I'm open for any discussions around GPCR projects. So, and to that, at this point, I finish my presentation. So that's me, and thank you very much for your attention, and I'm open for questions. Thank you.
Well, thanks a lot, Irina.